I've been successful, but more successful in terms of asking research questions, and it's been a difficult experience to answer many of the research questions, and I'm in the process of doing that. And I have a long last name, so it would be brutal to ask you to remember my name. So my students, they're all engineering students, like most of you. They call me as Dr. G, innovation, right? So that is what I call as innovation. But I'm from South Dakota School of Mines and Technology, and I'm fundamentally in the environmental engineering. I love environmental engineering, and I have a lot of passion for environmental engineering. But for my research, I always try to interrogate the questions at the interface of multiple disciplines: electrochemistry, microbiology, engineering, and then surfaces. So surface, we had a small debate, myself and Dr. Bhatti. So when I said Surface Engineering Research Center, he was thinking about surface water. And I was like stunned. Oh, is it? So it was kind of interesting to interpret the same word in two different meanings. And when I refer to surface, I'm talking about surface of the material. So I'm more interested in what happens when microbe sits on the surface and trying to answer some of the questions. For example, if I want to understand what interactions are helping microbes foul or form biofilm on the surface, could I interrogate those questions at angstrom scale or atomic scale, trying to get into the depth of what components of that particular material is contributing or helping the microbes to grow, and so on. So it's all going to happen at the interfaces, and that is the reason I framed the title as Understanding the Rules of Life in Biofilm. To be very specific, I'm talking about microbe sitting on the material surface, and if it is going to evolve as an adamant, as a stubborn biofilm, which is so difficult to remove, and in fact industry spend multi-million dollars every year to get rid of or to address the challenges associated with biofilms. And it's a very big research question. You need a lot of system biology tools and a lot of knowledge to get to know all about why microbes want to sit there, what kind of proteins they are going to express, and what kind of genes are going to be activated, what is the degree of gene expression, and so on. So I'm not alone. I have a very big team which is comprising of system biologists and many other people. And I'll talk about my team in a bit. But today, so this is going to be my agenda. I'm going to give brief introduction about the school, about what I do, about nanometer coatings. That's another fascinating concept. I got really fascinated, and I was introduced to the subject of nanoscale coating in 2010 when I was at Rensselaer Polytechnic Institute. So it's a fascinating concept in a sense that you'll not be able to see that material with a naked eye. For example, if I have this table and if I apply a nanoscale coating, you'll not be able to see that particular material on the surface. But what the scientific community claims is that nanometer coating has exceptional properties. When I say exceptional properties, I'm talking about something that would be stronger than steel, something that would be electrically conducting compared to copper, and many more. The list would continue to go. So I got curious in context of, could we use this particular nanometer coating to influence the biofilm formation and help some of the environmental engineering problems or some of the environmental problems and energy problems we are currently facing. So today I'm going to use three different examples, and we might not have time to get to that third and fourth, but we could do a good job in terms of second one. We are going to talk about sulfate reducing biofilms. So that's quite popular for civil engineers. And if you recognize something called as crown corrosion, sewer corrosion, something you might have heard in your wastewater treatment class, you are talking about microbes actually eating a large portion of the metal infrastructure. Believe it or not, the tiny microbe, they could make the huge holes, and then that could be responsible for deterioration or destruction of even the massive infrastructure, civil engineering infrastructure like bridges and so on. So that's sulfate reducing bacteria, and then geobacillus. So this is part of the NASA project we are currently doing, and we are trying to develop next generation microbial fuel cells. So for example, let's say we have a hypothetical lunar mission. Somebody or a group of people are planning to, visit moon, and they want to be in that mission for 15 years. They're going to generate a lot of this solid waste, and we are in middle of nowhere. Could we use this solid waste, generate electricity using the bacteria under ambient conditions? When I say ambient conditions, I'm talking about what we currently have in this room. And can we get the electrons out, meaning to say energy out, in form of direct current electricity? So 1.5 volt battery, the AAA battery you buy in Walmart is the form of direct current electricity. So could we generate that bio battery and harvest the energy and also store the energy and use that energy to drive maybe some kind of small scale devices when you are in the space missions? So that is the kind of question we are asking. And we are using GeoBacillus strain as the model biofilm because it is well versed, or it has capability to degrade even the worst or even the most recalcitrant form of the organic waste. You are talking about cellulose, semicellulose, and so on. And then finally, the last project we are talking about using the methane in the atmosphere, and we want to feed the bacteria with the methane and see if we could upcycle methane into some of the valuable products. So again, all at ambient conditions. And again, from research point of view, it's a very big research question. So I hope this is working. Yep. So that's South Dakota, if you haven't visited. And I'm sure most of you might have heard about something called as Mount Rushmore. How many of you know Mount Rushmore? All right. So that is what I use to make my school popular, Mount Rushmore. And we also have a fancy gold mine. It's an abandoned gold mine. We used to produce a lot of gold. That is the reason we call it as Black Hills, but there's no more gold left in the mines. And then a philanthropist and DOE, they spent a lot of money to convert this gold mine into a physics laboratory. We call it as Sanford Underground Research Lab. You could actually go several thousand feet deep into the earth, but in a systematic manner. You have elevators, and then you have this track, and there are kind of fancy trains that would run on these tracks. You can get to the point deep down the earth. You can collect the samples. We use this facility to do biofilm research, but the physicists, they would use this facility for something called as neutrino experiments. So something like containing the neutrinos within a given space and do research related to dark matter, which is beyond what I understand. But that is what we have in South Dakota, but I'm from South Dakota School of Mines and Technology. And as Dr. Bhatti said, I'm part of Surface Engineering Research Center. I'm talking about material surface. And we have state-of-art coating capabilities. And being able to deposit the nanoscale coating is one of the facility we have at School of Mines. And these are some of the ongoing research projects we are having at School of Mines, and most of them are collaborative research projects, except for the NSF Career, which is probably the only sole principal investigator award that is currently available in US. And most of the times, the National Science Foundation or any form of foundation, they are more interested in something called as convergent research, meaning to say a research driven by the problem statement, not by the scientific curiosity, but driven by the problem statement. And they would want to converge the experts from multiple disciplines. So all of the, or most of the projects that are being displayed on the slide, they represent multidisciplinary research projects, and we are talking about a group of 15 or 20 different professors. And for example, this particular track 2 bug remedy project, bug remedy stands for, it has a long definition, but we are talking about developing the infrastructure for the biofilm, and it involves experts from Montana State University, Oklahoma, University of Oklahoma, and South Dakota School of Mines and Technology. But from science point of view, what is more fascinating for me and what has been driving most of the research projects is, if I have a bacteria, let's say this is the bacteria, okay. The research question here is, if you understand the basics of microbiology, for bacteria to live, just like human beings, we need oxygen to breathe. No oxygen, we are going to die, right? And of course, microbes need food and we need food. The research question we are asking here is, you have the microbes, can you force the microbes to breathe in absence of typical terminal electron acceptors? Dissolved oxygen, something you use in the aeration tank, that's a typical form of the terminal electron acceptor. If there is no oxygen, it can live in anaerobic conditions. You could provide nitrate, sulfate, or some form of metals. And if you don't provide anything, it could live in the fermentation mode where carbon dioxide is used as electron acceptor. So what we are asking these microbes to do is, if this is the microbe, and maybe let's say this is a solid, okay, any form of metabolism is related to transferring the electrons from the redox cycle to the electron acceptor. So if there is no oxygen, this metabolism would shut off and then it can't live. So what we are asking is, here we come with a deal, we kind of come with an agreement with the microbe. We say, you have this particular environment, we are going to give you the food, but the only choice for you to breathe is you'll have to breathe against this solid surface, and we call it as electrode in the fuel cell literature or microbial fuel cell literature. And this particular solid is electrically conducting in nature. So we are kind of establishing an electrical communication between the microbe and the solid surface. If you are successful in doing that, what you are trying to achieve is getting the electrons out of the cell and driving a battery. That is one way of looking at this particular field. Or you can take the battery, get the electrons out, the electric current flows into the microbes, and you can use that to drive or manipulate the microbial physiology and derive products of your choice. You could produce hydrogen, you could produce methane or whatnot. So it's an evolving field of research. It's called as bioelectrochemistry, and that is what has been driving my research in this multidisciplinary research projects. So I'm slowly going, I'll slowly try to transition into the terms that I used in the title, extreme biofilms and then nanometer coatings. Extreme, how extreme is extreme? For example, if you put me, let's say, in a microwave oven which would produce like 40 or 50 degrees centigrade, probably I'll not survive for a long time, right? These microbes, they could actually grow at 100 degrees centigrade, boiling temperatures. We are using some of the hot springs in the Yellowstone National Park as the model sites. We have access to some of the sampling sites there, and we go get the biofilm samples at 60 degrees centigrade. It's kind of hard to believe, but they can grow at high temperature. Microbes, they have unique ability to grow in any given extreme conditions. Only criteria is you'll have to give it enough time for microbes to evolve, and the way of their survival mechanism is through the biofilm formation, right? So that is what is my definition of extreme biofilm. So you could talk about 150 degrees centigrade or 130 degrees centigrade, and you have some of the species living there. You can talk about minus 30 degrees centigrade, and there you are going to find some microbes which could tolerate such low temperature and could still live there. You can talk about microbes living under sulfuric acid conditions or acidic conditions, maybe pH of 2.5 and 3.5, and this is what has been discovered or explored in the past 50 years. We are not inventing something. All we did is we discovered that these microbes could actually live in those extreme conditions, and it could also live under salt conditions. So who cares, right? So we are engineers, and who cares if these microbes live in the extreme conditions? What are the implications? So from your day-to-day lives, what do you think are the implications if microbes can live under such extreme conditions? I usually tell when I teach a class, just like Dr. Bhatti or any other professor, when I teach class, I usually stop after 10-15 minutes, and this is what I tell to my students. I have a monotonous tone, and I want to break down my tone by asking a question.
Well, we could take like some of the tools that they've evolved to like
Sure. So that's a very good answer. You could actually manipulate or use the microbial physiology. For example, you're challenging these microbes to live under very stressful conditions, and therefore it has to trigger some of the special genes or proteins that would help the microbes survive under these conditions. And they're going to work probably, let's say, some kind of an accelerated rates, and we want to use that particular performance to drive some of the engineering devices. That would be a positive side, so you have a lot of positive thinking, keep it up. And on a negative perspective, it's also a challenge. For example, if I design a steel structure that would withstand, let us say, saline conditions in the seawater, let's say ship hull. So material scientists, they come up with a surface that can actually withstand such a high degree of salinity. And all of a sudden, you find out that microbes could actually live in those conditions, and they could actually accelerate microbial corrosion. So that's going to be a challenge. So you can use this microbial physiology both from positive perspective, but it's also going to induce challenges. So this is the schematic of the Stanford Underground Research Lab I talked about, and you could actually go down, deep down 2.5 kilometers, and of course 2.5 kilometers would be an exaggeration, because they would not, you have the access, but they would not let you get to that deep region due to the safety concerns. But you could definitely get to 1.5 kilometers. You have these nice shafts, and you have some of the air-conditioned rooms downstairs, and you can, take breath for some time and then go back to the field and do all kind of things. But if you ever come to South Dakota, this is in Lead, and you are more than welcome to visit this particular facility. So this particular site is what would simulate the hot temperatures, and what we are doing is we are using this particular facility to simulate something called as microgravity, or the harsh environments experienced by the in the space. So there is a special field of research called as astrobiology, and what they do is they want to simulate what life would exist in the outer space. And of course, I can't go build my house in the outer space, and I can't do my research in the research lab. So they use this particular facility to simulate what would happen in the outer space, and that is how they deduce some of the findings related to life in the outer space. So these are some of the sampling sites we have that we could access at Yellowstone National Park due to our collaboration with Montana State University. And Yellowstone National Park it's a feast to eyes. It's very colorful, and you can see a lot of fascinating and beautiful, pretty sites. And what imparts color to these sites is the microbes. There are different types of microbes that would exist at different layers, which would vary in type of temperature, and that is what would impart color. But we are more interested in the physiology of the microbes living under those extreme conditions. Now I'm going to introduce a rare combination. Now we talked about extreme biofilms that can withstand extreme conditions, and we are also talking about extreme conditions that would degrade any given material. For example, if I put a piece of paper or polyester in the boiling water, it's going to degrade no matter what you do, right? So we are looking at combining the nanometer coating, something you can't see with the naked eye, and we want to study the interaction of this nanometer coating with respect to the microbes, which could live under those extreme conditions. And the research question is, can you design nanometer coating to help or to manipulate the interaction between the microbes and the materials, and use it as an advantage to design better engineering applications, is what we are trying to interrogate. So if you want to visualize, I'll not get into the depth of materials here, but if you want to visualize what is nanometer coating, take a piece of coal. It's amorphous substance. It's all made out of carbon. It's made out of carbon atoms, but it's typically arranged, if you just imagine one slice of the entire carbon or the coal, it's all going to be based on these carbon atoms connected through covalent bonds. But in reality, this layer would not exist as an independent layer. It's always going to be hundreds of layers or maybe tens to hundreds of layers. A scientist, out of his own curiosity, he developed a method to peel off one layer, something you can't touch with the naked eye. He peeled off that particular layer, and he was able to isolate it. But who cares, right? If you isolate one layer, two layers, it's not a big deal. But what was the big deal was they proved that if you isolate this particular layer, since you have only one layer of carbon atoms, the electrons are going to transport in much faster rate. And the light would go through or penetrate through this particular layer in much faster rate. And the list would continue. We are talking about strongest material, and I think I have a slide somewhere I'm going to talk about it. So again, it's probably an over-exaggerated site, but a good visual to help you understand the exceptional qualities of these particular layers. Some professor came up with this schematic. You take this nanometer coating, and if you have the ability to hold that nanometer coating, you get this elephant, and I can't lift the elephant. Probably we need hundreds of people to carry the elephant. If you put that elephant on this nanometer coating, the coating would not break, is what is being presented. And that is what is the strength of this material. And I'm not going to quantify these properties here, but there is literature which proves that it's as strong as steel. And if you take any coating, it's mostly brittle. For example, if I want to bend this thing, it's not going to happen, right? But you could actually use this material to form a flexible coating. And they're using this particular feature to develop a thin, non-invasive watch that would embed or incorporate all of these chips. And that is how you have this foldable watch made out of this graphene material. In future, you could see foldable televisions. For example, you might have television, and then I want to carry the television in my pocket or maybe in the car. I'm going to fold it into a cylinder, something like what you would do with the paper, and you would still be able to run the television, unwrap it, and watch the screen. So that is the kind of technology we are talking about, and that is the kind of future we are envisioning using these nanometer coatings, right? But I'm not a physicist. I'm not into field of semiconductors, but my interest is more in context of using these materials for biofilm applications. Just imagine that you have a steel surface, a huge bridge. Could I put this coating and develop an everlasting coating that would prevent the degradation of the material? It's going to happen. No matter what you do, you have the steel structure. The typical practice is to put the zinc coating, and it could last, or it could offer the protection for 20 years. But beyond that, you'll have to service, put additional layer of zinc coating, and so on. So we are talking about using nanoscale coating, and could we minimize the corrosion or the oxidation of the given metals is the question we are trying to ask. And graphene is one example, but there are 700 different nanoscale materials that have been isolated in the past not 20 years, but 10 years, or maybe 8 years. And out of these 700 materials, only few have been synthesized in the lab. Some of them have been simulated, but we are yet in process of simulating, or yet in process of synthesizing that particular material. And the way you do this is, if you have scientific curiosity, and this is how they invented the graphene, they took the Scotch tape, put it on the coal or some kind of carbonaceous material, peel it off once, take another Scotch tape, peeled it continuously, and then they isolated one single layer of graphene. So today I'm going to talk about graphene coming from graphite, and boron nitride coming from the bulk powder of boron nitride. But whenever I refer to graphene or boron nitride, I'm talking about one sheet or one layer of well-connected atoms in a systematic manner, and that is what is going to give the definition of crystal. These are all crystalline materials. So let's look at the microbial corrosion problem. And I call it as a $1 billion problem, because that is what we spend every year. Take Department of Defense. Take an example of F-16. or take an example of ship hull, or take an example of steel bridge. Whenever you expose the metal to the atmosphere, and especially in presence of moisture and the microbe, you are facing something called as a microbial corrosion problem. So if you take this steel bar, it's made out of mild steel, you expose it to the microbes, and glutaraldehyde is a type of biocide used to inhibit microbes. People thought that if you use biocides, it's going to kill the microbes. It might happen momentarily, but eventually the microbes will get used to the biocides and they gain resistance to the biocides, and this is what has happened in presence of the sulfate reducing bacteria, such a huge hole. If you give this to my hand, I will not be able to break it, but microbes could put a large hole, break it apart, and do all kind of things. The only reason why they would do it is to gain the energy from the oxidation process. So it's all about their survival. So you're actually, if you want to protect this material, you're actually challenging the microbes to stop their survival mechanisms. So survival of the fittest, they want to live, you don't want them to live, so it's going to be a large battle, right? To get you excited, and this is not mine, but my grad student, he came up with this article somewhere from literature. It's a Titanic ship, and it's buried deep down in the water. Based on the studies, the wreck of the Titanic ship would be depleted completely by 2030. Microbes, they are living in the deep water, those are saline conditions. People thought that microbes would not live under saline conditions, but they always evolve, they constantly evolve, and they learned a mechanism to overcome something called as osmosis effect due to the gradient in the salt concentration, and now they are happily using this metal as the source of energy, and eventually the fate would be the disrupted or destroyed or dissolved or eaten away components of the ship. So again, I'm not going to get into the depth of biology, but these are different stages of biofilm, just like human beings. You know, you are born as a baby, grow up as a kid, teenager, middle-aged, and so on. Bacteria, they find the surface and they kind of clean the surface. They spit something called as proteins and carbohydrates, and they condition the surface. Once they condition the surface, the cells are going to go attach to the surface. They are going to spit another layer of polymer, and this polymer is called biofilm layer or epoxy polysaccharide layer, and it's almost like a house, a very beautiful house, and it's a very strong house. Microbes are going to live in the biofilm, and once it forms biofilm, then it's very difficult to disrupt the biofilm. You can use the most powerful biocides, you can use the most powerful water flow you can think of, but you'll not be able to wash off that biofilm onto the surface. So that would be the degree of maturation the biofilm could achieve, and once it gets to that point, they are going to be 100 times more powerful compared to the suspended form of microbes, and the degree of corrosion would be 100 times or 100-fold higher. So if you can understand what is going on at different stages, especially at the earlier stages, for example, if you could do something to minimize or discourage the attachment or the conditioning process right at beginning, it's going to happen after 1 or 2 hours, and if you could understand that from system biology level, you are going to get more permanent solution compared to currently available engineering solutions of using biocides or using coatings. And there are 3 different types of microbial corrosion. The microbes can actually oxidize the metals, get the electrons out, and use the metal oxidation as source of energy. So that's one form. Second, they are going to secrete the acids, and acids are going to corrode the metals, and that's second type of corrosion. But if you have the plastic material, plastic is not a metal, but it has the carbon and hydrogen bond, they could also use the carbon in the polymers as their food, and that's another form of corrosion. So we are interested in all the 3 types, but as engineers, you'll probably be more interested in the polymers, because most of the coatings we currently use are polymer coatings, epoxy coatings. If you go talk to a technician in the wastewater treatment plant, they are going to tell you that, hey, I coat the surface of the activated sludge process or the aeration tank with something called as polyurethane or epoxy material. They're all based off polymers, and they're all good sources of food for the microbes. So these are some of the reasons why we should, or why I think, or why we think we should explore the next generation coatings based on the nanometer graphene or hexagonal boron nitride. And I'm not going to read through all of this list, but again, as I said, they could serve as the food, and they are porous coatings, meaning to say the salt can intrude through the pores and then disrupt the metal surface. So if I have the metal, I'm going to protect the metal by depositing a coating, and I want to hide the underlying surface from the microbes. We call it as a barrier property. So to overcome this, the research question here is, could we develop high performance coatings, non-invasive coatings? I'm calling it as non-invasive coatings because if I have, let's say, 1 centimeter thick, let's say, copper surface, and if you add another centimeter thick, let's say, polymer coating, the thickness is comparable to what you have beneath the surface, right? So here I'm talking about angstrom scale coating, and therefore it's going to cause minimal disruption to the bulk properties of the underlying surface. So my hypothesis is, could I slow down the corrosion? You'll never be able to prevent corrosion, that's impossible, but you could slow down the corrosion. And my question is, could I use this nanometer coating, in this case boron nitride or graphene, to slow down the corrosion of the copper? And we are kind of correlating the parameters from 2 different components. You have the system biology components, and we have the material properties. And material properties more at the interfaces of the nano scale. So the questions we are trying to ask is, if you apply the coating, if you modify the properties of the coating, modify the properties of the bulk material using the coating, and if you can develop a list of parameters which would signify the material properties at the nano scale, for example, the surface roughness or the number of layers of these nanomaterials, or it could be surface charge, or it could be wettability or hydrophilicity, hydrophobicity. Could you correlate the impact of each of these material property with respect to the biofilm phenotype? Phenotype is about how you look. For example, why is my hair black, and why am I talking the way I am currently doing? That is phenotype. And gene or genotype is what genes are responsible for my action. So those are genotypes. So my question here is, could we correlate the surface properties with the biofilm phenotypes? And could I also get into the depth of finding out what genes are being activated or what genes are being expressed with respect to change in the surface property? So that's a very, very difficult research question, and also very difficult to do something in the lab. But that is our overarching research question. And we are using something called as sulfate reducing bacteria, D-sulfo vibrio alaskensis strain G20. So I'm going to use the acronym G20 throughout the presentation. And this is the schematic of the microbial corrosion cell my grad students are using. And if you like to zoom the images, you could actually use a transmission electron microscope, and you can zoom it million times. And if you do it for this particular bacteria, this is what you are going to see. You'll be able to look at the outer surface. You'll be able to measure the dimension. It's approximately 0.5 micron in diameter, and you'll also be able to see what is going on within the cell. So it makes everything transparent without damaging the sample. And we are using G20, and we are using a batch reactor, and we are using all kinds of characterization methods based on microscopy, spectroscopy, to really tell that you are dealing with nanometer coating. For example, if you give me a steel coupon, and I'm going to say that I have the graphene coating on the steel coupon, but what is the guarantee? How do I prove that I have a coating on it, right? So you'll not be able to see that with the naked eye. So that is one thing. And second thing is you might have the coating, but how do you prove that what you have is what you want to have? How do you prove that it is graphene? So that characterization itself is another challenging task. And without getting into the details, we have the optical microscopy, the images from SEM, not SEM, but TEM, Raman spectroscopy, and this is the SAED pattern coming from the TEM. And it's difficult to see, but if you look at these two lines on the TEM images, those two lines are talking about the distance between the two layers of the carbon atoms, and this is what we call as bilayered graphene or hexagonal boron nitride material. So this is some evidence, visual evidence, to prove that hexagonal boron nitride is more effective compared to graphene coatings to protect the microbial corrosion of copper. And on the left hand side, the panel shows that no matter what coating you are going to use, you can't stop microbes from forming biofilm. They are going to form biofilm, and they are going to sit on the top of the surface. But only thing they are going to do is they are going to hide the surface from the biofilm on the top. If it could do it for a long time, we are saying that it is going to offer barrier properties. So that is what is happening here. And as you see, the boron nitride is offering better corrosion resistance compared to copper, which is controlled, which is kind of expected. But what is unexpected is the graphene coating, instead of slowing down the corrosion, it's actually aggravating corrosion. It's working in counterintuitive manner, and it's kind of a complex graph here, and it's based on the electrochemical impedance spectroscopy. Let's not. So, in the very short message from these graphs is graphene is aggravating corrosion, it's enhancing corrosion. Hexagonal boron nitride is minimizing corrosion. Okay. So again, to break down my monotonous tone, why do you think a coating would accelerate corrosion or enhance the corrosion process? Do you believe me? Yeah.
If it were a kind of a accelerant, almost like a catalyst for the bacteria, it could, instead of impeding or slowing down the corrosion rate, or
sure,
giving something else to feed on, I guess, like you were saying.
Yeah.
The catalyst would almost make them hungrier, I guess.
Yeah.
If you will.
Yeah. So that's, you are thinking along very good lines. I think you'll become a scientist in future. Yeah, so that's a good thought process, but we had to think for a very long time. You know, we are talking about adding a barrier. Why is this coating promoting corrosion when everybody is claiming that? Yeah.
Does it help them like adsorb to the material better? So it just has
That's a possibility. That's a possibility, and that's one of the challenge that all of us or most of the engineers should be aware of. There are certain material properties which would actually promote the attachment of the biofilms, right? So those are all good thoughts, but what we found is the major reason is it gets into the depth of the chemistry or corrosion. There is something called as galvanic corrosion. How many of you did something called as a potato experiment when you were in high school? Okay, you did a potato experiment. So could you explain what is potato experiment? Right. So what's your name?
you're talking about where you use the potato and you
Yeah.
and you essentially
Sure.
generate a current from it.
Yeah, I don't know your name, but he remembers the experiment very well. You take the potato, you put two metals. We are talking about two dissimilar metals. And whenever you have dissimilar metals, they enjoy facilitating the flow of electrons. They want to achieve steady state. For example, it doesn't happen with the human beings. If I have $1 million, I don't want to give you my $1 million, right? But metals, they are more collaborative in nature. If they are in different state, we use the term called as redox potential. they always try to achieve a steady state condition by exchanging the electrons, and that principle is called as galvanic corrosion. You can take a zinc metal and you can take a copper metal, and you're going to see that if you use the multimeter, you're going to register significant potential difference between these two metals due to their inherent state of electronic configuration. The same exact thing, graphene, as I told you, is super conductor of electricity. Electrons would love flowing through the graphene at a very fast rate. We measure that in terms of electrical conductivity, electron mobility, Hall quantum kinetics, and other things. And then you have the copper. So you have the copper at the bottom and graphene at the top. So copper and carbon, one is metal, one is kind of a semi-metal, and there's going to be a potential difference between these two. And we talked about microbes. They're going to enjoy, and again, it's all about their survival. They're going to enjoy extracting the electrons, and then therefore the energy out of these two metals, or difference between these two metals, and use it as a source of energy. So it's kind of promoting the galvanic corrosion, and the reason is because graphene is electrically conducting material. So, and in terms of the lab, it's very difficult to implement the experiment even to prove that it causes galvanic corrosion, because the schematic is pretty, but you're talking about transferring that coating, nanometer coating. How would I get the how would I peel the coating and put it use it as the counter electrode or working electrode? It's very difficult. So all I can do is I can take the nanometer coating and I can transfer it onto the other substrate. And in this case, we are using silicon wafer, right? And if you enjoy scientific curiosity, it's very challenging experiment, but also very interesting experiment. And again, this is the graph, and the graph tells you that the graphene, this particular line, is at much higher range compared to the copper or even hexagonal boron nitride, meaning to say generating more of the galvanic corrosion in terms of galvanic current. And all the subsequent slides, I don't know how much time I have. Oops. 5 minutes, that was pretty fast. I did not even get to 23rd slide. So let me operate this in this mode so that I can skip some of the slides and move on to others. So that was the finding, and then we are getting into the depth of, so since graphene coating is not going to work, one research question we are asking is what can we do to make these graphene coatings work? And one way would be to add more number of layers so that I don't have any defects in the coatings, and therefore prevent some of the electron acceptors from seeping through the pores. For example, if I could prevent oxygen or protons from getting to the bottom, I'll be able to decouple the galvanic corrosion or decouple the galvanic reactions, because the galvanic corrosion is because of the electrochemical reactions happening on the working electrode and the counter electrode. So if I can shut off the reaction, you are also able to minimize the corrosion. But we are getting into the depth of also the hexagonal boron nitride, because it's an insulating coating. So that is the difference. Both of them are alike in terms of how they look. They look like chicken ring. So 6 carbon atoms or 6 different atoms arranged in a hexagonal pattern. And one is conductor and one is insulator. So we are taking advantage of the insulating properties of hexagonal boron nitride coating. But my ultimate objective is to map the surface properties with the biofilm phenotypes. So we came to the point where we are quantifying the biofilm thickness, biofilm morphology, and we are getting into more excited stuff. This is carried out by a biologist in my lab. So we are looking at what genes are being expressed. So A, T, G, C, those are 4 different amino acids, and the combination of these 4 amino acids is what is going to define your gene or the gene in the bacteria. So we want to identify what genes are responsible for the microbial corrosion, and we want to identify what conditions would overexpress some of the genes which are potentially responsible for microbial corrosion. If we have that fundamental knowledge, then we can go back and design better materials, manipulate the surface properties that would help you control microbial corrosion or any other problem statement of your choice. I'll spend 2 minutes each on 2 of the projects. Is it okay, Dr. Bhatti? Okay, so Dr. Bhatti approves and he agrees. So I am going to do that. So 2 minutes each, and leave adequate time for some questions. So this is my pet project, and it's funded project. When I say pet project, it's not a Hobby Lobby project. It's funded by the NASA IPSCOR. So it's a $1 million project, involves 5 or 6 faculty. And the question we are trying to ask is, we have the waste, we have these astronauts, or the crew, let's call them a space crew. They're going to generate 2.2 pounds or maybe 4.5 pounds of solid waste on a daily basis. It could be food that we eat, leftover food waste, or it could be wrapped paper. And there's also interesting form of solid waste compared to what we generate on planet Earth, and it's called as diaper. Astronauts, they wear diapers. Those are probably fancy forms of diapers, and they are made out of very fancy materials, synthetic materials, and they're very hard to degrade. So we want to find out whether there exists a robust biofilm that can degrade all of these components very successfully at high temperatures without promoting the growth of pathogens, because pathogens is a significant issue in the space missions. And once you get to that point, can I extract the electrons out? If I don't do that, then what is the point of this technology, right? So extract the electrons out, harvest those charges using the supercapacitors, graphene supercapacitors, and then demonstrate whether we could use this energy to maybe drive a micro rover or maybe small bot on the space and use it for sensor application or other applications. So that's kind of a long-term question we are trying to ask. And we have some preliminary data here. We found that the Geobacillus species that we are working on could eat or live on the typical pure substrates. For example, if you give me a candy bar, a Snickers, I'll be happy, right? I'll be jumping with joy. And that is what is being represented by the glucose. If you give me grass, or maybe if you give me broccoli, probably I'll eat it. But if you give me grass, I'll not do it. But you are actually asking microbes to do the same thing. Ask microbes to eat corn stover and also the simulated space waste, which would have some kind of polymer components. But so it could do it to some extent. And we are also developing the graphene electrodes. So that's what I told you, right? I want to see how microbes would interact with nanometer coating. So one of the experiment, we took the nickel surface, highly conducting surface, and we added graphene onto the nickel surface. And when we looked at the performance of a graphene coated nickel is probably 10 or even 100-fold higher compared to the control, and also compared to any of the commercially available electrode materials. So we are getting into the science. We are getting into so we have the performance, but we are trying to find out why it is happening. And then we have this series of slides which will take you into proteomic analysis, RT-PCR analysis, transcriptome analysis, and we are trying to get into the details of what genes are being expressed, why they are being expressed, and how would the electrons flow from within the cell to outside the cell through the cellular interface. So since you have 5 minutes, you are fortunate that I'm not going to bore you to death by talking more details. But this might be of your interest. Most of you are engineers here. So this is what we want to develop. We want to develop a stack, put all of these biomodules together in a series or parallel configuration. So instead of getting 1 volt, can I get 100 volts is my question. Instead of getting 1 microamp, can I get 10,000 milliamps or maybe even 100 amps, which is what would drive any of the electrical device. For example, if you take my laptop charger, I at least need 20 milliamps of current. If I don't do that, it's not going to charge my laptop, right? I might have flow of electrons, but it's useless. So that is what we are trying to ask. And the last project we are trying to do, as I told you before, is grow the biofilms which are capable of using methane, which are capable of oxidizing methane. If you just use a chemistry approach, you'll need a lot of catalysts, very high temperature, 300 degrees centigrade, 500 degrees centigrade, to oxidize methane and to generate electricity or generate any products. We want to develop biological routes where we could use microbes to convert methane into useful products under ambient conditions. So that is the application part, but we are also interested in the fundamental research questions part, and again we are using the system biology tools. And we have collected the samples, we are in process of isolating the microbes, and in conclusion, we are studying the rules of life on how the microbes would form biofilms on these surfaces, engineered surfaces, with respect to changes in these surface properties. I have a video, but I'm not going to play it, but this would be time for you to answer, for you to ask questions, and for me to take opportunity to answer your questions. Yeah.
For the space exploration, you were saying that certain genes or certain types of bacteria or certain parts of bacteria biodegrade the polymers better. Could you, and you were saying earlier that you can manipulate the bacteria such that you get the best qualities out of them.
Yeah.
Through kind of like you were saying, I guess in the mind where you do all the
Sure.
manipulations with the
Sure.
bacteria. Could
Yeah.
you use those manipulations to be advantageous towards?
Exactly. So that is what we are trying to do here. But I would rephrase those are very interesting thoughts. But I would rephrase your statement slightly. What you could do is you can manipulate material properties, and what you could do is you could expose these microbes to different conditions. But we are not. You have no ability to manipulate the microbial physiology, but you can wait until the microbes evolve to sustain those conditions. However, you could also manipulate microbes when you use something called as gene knockout studies. You could put genes of your choice into the microbes, and you could try to achieve the kind of biofilm phenotype you want to achieve. And there's something called as CRISPR technology. So it's a debate. It's a hard debate. People in China, what they did is they used CRISPR technologies to actually insert some of the genes into the human baby, and they were able to manipulate some of the genes. So now it became a question of ethics, and there's a lot of debate going on. But in simple answer, yes, you could actually do it in both ways. You're right. Yep.
To counteract some of so you said that microbes will always evolve to be able to degrade structures like steel bridges and stuff.
Mhm.
Could you use directed evolution techniques to put them in only give them steel or whatever the substance is,
Mhm.
and see what they'll evolve to come up with to degrade it, and then use that knowledge of what they will do to counteract it in the future?
Sure. You have a lot of scientists in your room. That's in fact the exact research direction that's kind of happening. They call it. I mean, I don't deal with that subject, but it's called as directed evolution approach. So microbes, they evolve, and specifically the enzymes within the microbes, they evolve as function of time. So scientists are coming up with approaches where they could accelerate the evolution process. For example, if the evolution is going to take millions of years, could we do it in maybe a few minutes or few hours is the research question. And that person, along with 2 other people, they just received Nobel Prize last year. They call it as phage assisted directed evolution. So had you thought about this idea maybe 10 years before, you would have won the Nobel Prize. Yep, that's a good question.